Hello folks, now I'm introducing a research finding we recently published on iMeta. The title is Dietary Fiber Chemical Structure Determined Gut Microbiota Dynamics. Dietary carbs in our food are super different, and our genome encodes only 17 digestive enzymes for lactose, starch, and sucrose, but none for dietary fibers. To compensate for this catalytic deficiency, humans rely on their gut microbiota, which can encode proteins designed to recognize and consume dietary glycans. Different dietary fibers could induce different gene expression in a bacterium for its utilization. Therefore, precise supplementation of dietary fiber is a promising intervention for gut microbiota-related health issues. It was initially proposed that the dietary fiber chemical structure was related to the gut microbiota dynamics. Thus, certain chemically structured dietary fibers for specific gut bacteria could reach predictable gut microbiota modulation. Although there are some preliminary reports on the relationship between dietary fiber structure and gut microbiota dynamics, the detailed mechanism behind them remains largely unknown. Here, we combine in vitro and in vivo bacterial dynamic studies with inulin or AssP as the sole carbon source and a bacterial genome analysis to identify the relation between dietary fiber chemical structures and gut microbiota dynamics. We have initially analyzed the monosaccharide composition of AssP, and here we further determined its structure, especially the glycosidic linkages, by a combination of 1- and 2-dimensional NMR spectroscopy method. It's known that inulin is a beta-2-1 glycosidic bonds linked to fructose. Here we identified AssP as alpha-1,2 glycosidic bonds linked to glucose and fructose, beta-2,6 glycosidic bonds linked to fructose, and other bonds linked to galactose and arabinosis. In the following, probiotic bacteria were selected and cultured in a medium where either inulin or AssP was offered as the only carbon source, and each bacteria's growth was monitored. Additionally, in vivo experiment was performed by providing C. elegans with either inulin, AssP, or ASS on the basic NGM as a dietary fiber supplement. The gut microbiota of C. elegans were then sequenced. In vitro growth experiments show that all probiotic bacteria could grow in the presence of either inulin or AssP with different stationary biomass and growth rate. However, when AssP was used as a sole carbon source, probiotic bacteria yielded higher biomass and faster maximum growth rate than inulin. These results indicate that AssP supported more bacterial growth than inulin in vitro. Then the growth patterns of different probiotics with different carbon sources were analyzed. We found that these probiotics clustered separately once inulin was used as a sole carbon source. Contrastingly, clustering between all probiotics became indistinctive once AssP was used as the sole carbon source. These results indicate that AssP catered to more probiotics in their growth. Then we analyzed the whole genome of all probiotics to find the relation between them. We found that for every bacterium, more genes support bacterial growth on AssP than inulin as a sole carbon source. In addition, different bacteria can utilize different glycosidic bonds. Similarly, a monosaccharide connected via a certain glycosidic bond could meet the genetic requirement of being hydrolyzed by one bacterium, but not necessarily another. In vivo experiments show that worms fed with AssP exhibited a significant increase in gut microbiota richness, while worms supplied with different dietary fibers exhibited relatively stable microbial diversity and evenness. Beta diversity indicated an almost overlap between worms fed on the base medium and those with inulin supplementation. The bacteria composition at the odor level is also similar in worms fed with base and inulin. Network analysis indicated that in worms supplemented with ASSP, more interactions between bacteria increased, and they were all positive, which showed that ASSP could induce more complex worm microbiota growth. When we compare the in vitro probiotic growth and the in vivo worm gut microbiota dynamics supplemented with different dietary fibers, we found that only in worms supplemented with ASSP, all geners which we used in the vitro experiments increased. That is, ASSP has a better probiotic effect. In conclusion, genomic analysis of individually cultured bacteria indicated that the more complex the dietary fiber utilized as a carbon source, the more bacteria it could possibly support. And this dietary fiber chemical structure bacterial growth relation is validated in the C. elegans animal model, provided that the microbiota community interactions and gene transfer scenarios are not considered. This study lays a theoretical foundation for the food, nutritional supplements, and medical intervention of gut microbiota.